so subdued this morning? Do you have any questions? Okay, we were in the midst of talking about virulence factors, and we've covered previously the exoenzymes, and now we're taking a run through some of the toxins that pathogens can produce, another virulence factor. And remember from that list, there are quite a few different modes of action that toxins can take. The one that we're talking about right now is the cholera toxin. What's the kind of overall characteristic of the disease cholera? Well, dehydration is a consequence of massive water loss of fluids to the intestinal lumen, causing dehydration, drop in blood pressure, hypovolemic shock. This particular toxin is an enterotoxin. What does that mean? What's its target tissue? Got epithelial tissue. So on this slide up here, you can see that the enterotoxin, which is an AB toxin, has to be able to bind to receptors on the surface of these epithelial cells. So this is more than likely an intestinal cell. What happens then is that particular toxin is brought in by endocytosis, and the vesicle then that contains the toxin travels to the endoplasmic reticulum, and basically then the toxin is separated so that its two components have different fates. Okay? The B component is exported back to the cytoplasm, and the A component, the active component, ultimately will bind to a GTP ACE protein. G proteins are very important signaling proteins in eukaryotic cells, and the ultimate effect of this binding of the toxin to the G protein is an increase in cyclic AMP activity. Remember, cyclic AMP is the second messenger, and that particular second messenger can result in a variety of physiological consequences. In this particular case, it activates transport systems, especially these chloride and potassium channels in the membrane. And as we start pumping out chloride and potassium ions into the lumen of the intestine, what follows? Water, osmosis, okay? And so water starts rushing out of the cytoplasm of the cell into the lumen of the intestine. As I said, this particular toxin is not as common in developed countries now that we have good sanitation systems. Unwittingly, there is a maritime practice which has been responsible for transferring bacteria, the Vibrio, that produce the toxin from one country to another during the shipping of goods. When you unload a big shipping vessel at a port, all the weight goes off, and that weight is your ballast. And so one of the things that they'll do if they're not picking up another set of containers to go to the next port, then they will fill the hull with water, seawater, for ballast. Okay? And then when you get back to your next location and you want to take on more goods, you discharge that seawater ballast into the bay of the country that you're docking at now. And if that water is contaminated with the Vibrio bacteria, then that can transmit that contaminated water from one port to another. So some kind of interesting, human-made systems actually contribute to the spread of this particular disease. Another toxin that we talked about that I won't go back over again is the C. diff toxin. C. diff, remember, is a toxin produced by the Clostridium difficale bacteria. And that's the one that about 5 to 10% of us carry around normally in our gut. Why doesn't this toxin cause us a problem? Because the C. diff bacteria are held in check by our normal flora. About 70% of young kids have C. diff in their gut. Part of the reason that it's so much more predominant is they haven't quite developed yet their really robust microbial flora, and consequently it is not held in check as much as it is in an adult. And this C. diff toxin can have various effects on cells of the intestine. It can break down the connections between intestinal cells, so things start leaking. It can kill actually intestinal cells, and you can get some necrosis. There's a disease called ulcerative colitis that is sometimes the result of the C. diff toxin. It's a pretty nasty disease. Do you remember what we said primarily was cause of it? It's normally held in check by your normal flora. Yep, yeah, particularly IV antibiotics in the hospital. Okay, so this is a situation where you've got to be really careful once people come up with this particular disease. Some of the treatments for this are discontinuing use of antibiotic, but obviously you were taking the antibiotic for some condition to begin with, so that can be problems. Some people who suffer from some of these long-term inflammatory bowel diseases, and C. diff is an inflammatory bowel disease. Crohn's, have you heard of Crohn's disease? Crohn's is an inflammatory bowel disease. We're starting to use more and more probiotics in those particular kind of conditions. You know what a probiotic is?
Good bacteria.
Good bacteria, yep. Good life, bro. Yeah. You can take probiotic in pill forms, and one of the things that they're looking into now is doing fecal transplants from people with very robust, good bacterial populations in their feces. And the fecal transplants can sometimes either be done kind of surgically almost by injecting material, or in pill form, popularly known as poop pills. Okay? But restoring your normal flora on kind of a lesser scale, sometimes it's recommended that if you've been through a long-term antibiotic therapy, that you start eating yogurt, because yogurt's got some active bacterial cultures. But the probiotics are kind of a more diverse group of microorganisms. The thing you primarily find in commercial yogurts are lactobacillus bacteria, but the probiotics or the fecal transplants contain a much more robust group of microorganisms. And so this is something that kind of grosses people out, but if you suffer from one of the inflammatory bowel diseases, this isn't looking so bad, okay, because those are really very serious and debilitating types of diseases. Okay, we're going to take a look at two diseases that are related, even though they have different symptoms, different microorganisms cause them, but basically they're related because they're produced by the same basic type of toxin. So there is a botulism toxin and a tetanus toxin. But really, they both work pretty much the same way. So the question is, why do we get such different outcomes from basically the same toxin and have two separate diseases? Let's start out with botulism first. Know anything about botulism? Heard of it before? Yeah, botulism toxin causes flaccid paralysis of the muscles. And that can include the diaphragm, the intercostal muscles of your rib cage, So if those muscles can't expand and contract, what's going to happen to you? Can't breathe, okay? So one of the treatments for botulism poisoning is putting somebody on a mechanical ventilator and then treating with antibiotics, for example, and hoping that the ventilator can keep you stable while the antibiotics are doing their work. Anybody know how you primarily get botulism? What's one of the common ways to get botulism? Food? Improper canning of vegetables is one of the primary ways in the past that people have gotten botulism poisoning. The botulism toxins produced by the bacteria Clostridium botulinum. What should you be thinking about when you think Clostridium now? What kind of an organism? From gangrene, we talked about Clostridium perfringens, and that was an anaerobe. Clostridium bacteria are anaerobes. That's what we had growing in our gas back jar as the positive control for an anaerobe. So what happens? Well, remember I said Clostridium were oftentimes found in the soil. And let's say you pick some green beans from your garden. There's probably going to be some soil. There's probably going to be some spores on those green beans. Okay. Well, if you just cook them up and boil them, then the temperature, I think you need to get about 82 degrees Celsius to inactivate the botulism toxin. Or if you boil long enough, you'll kill most of the spores. That's typically pretty good. But if you want to can them, what you do is basically you autoclave them. You put them in a pressure cooker. And during the autoclaving process, oxygen is driven out of the containers. And if you don't autoclave or pressure cook for long enough or to a high enough temperature, then the spores can survive. And now they're growing in they're in this nice anaerobic environment, and the spores can begin to germinate and produce the toxin. When these cells are growing, they're obtaining their energy by fermentation. And again, what does fermentation oftentimes produce? Gas. Okay? So have you ever seen a can that is swollen, almost ready to burst because of the gas that's built up in it? That's probably because of Clostridium botulinum, its presence in that. So you never want to eat vegetables from a can that looks like that. It's a really potent toxin. In fact, it's probably one of the most potent neurotoxins that exists. There's some concern about this from a bioterrorism point of view, because it's estimated that 30 grams of this could be enough to kill everybody in the United States. That's a really potent toxin. So what's it doing? How does it result in this flaccid paralysis? Well, basically what happens is this particular toxin blocks acetylcholine release at the myoneural junction. So up here, we've got a nice little motor neuron. And here's a muscle that motor neuron innervates. So here is the myoneural junction. When that motor neuron receives a stimulus from somewhere in the nerve net, then vesicles that are in that motor neuron travel to the membrane, fuse to the membrane, and then release that particular neurotransmitter, acetylcholine, and that causes muscular contraction. So the botulism toxin interferes with that release of acetylcholine. How does it do it? Well, it's a peptidase actually, and it cleaves one of the proteins that's needed for those vesicles to be able to fuse to the membrane and release the neurotransmitter. Food botulism is not the only type of botulism that we can see. There is another type of botulism called infant botulism. This results when an individual, a young child, ingests the spores of the Clostridium. And when those spores get down into the large intestine gets a little bit anaerobic down there. You've got a fairly diverse population of anaerobes living in kind of the recesses of your gut. Well, if you ingest those spores, and even if they germinate, normally your flora, your normal flora, will outcompete the Clostridium, just like it is the Clostridium difficile. And it doesn't cause you a problem. But infants, because they're still developing that normal flora, if they ingest Clostridium botulinum spores and they germinate, then they can cause the disease because the bacteria produce the toxin. Why might infants ingest spores? Well, one of the foods that oftentimes contains Clostridium botulinum spores is honey. And so one of the recommendations pediatricians will give you if you have children is no honey for a child under the age of 1. Until you get to be at least 1, your intestinal flora just hasn't built up enough to outcompete any of those organisms that you ingest. There is also wound botulism, where Clostridium botulinum spores are introduced as a result of a wound. So that makes it, in some respects, kind of like the gangrene almost. Okay, there's got to be some kind of trauma first, and then that creates an anaerobic pocket in the dead tissue where the spores could germinate. What else do we use botulism toxin for, medically? Botox. Okay, how does that work? It causes paralysis of muscles. Actually, facial wrinkles are the result of skin muscles contracting. And so if you can get small doses, that causes those skin muscles to relax, but it's not enough of a dose to be systemic and cause problems with your skeletal muscles. They're also using Botox now a little bit in treating migraines. Okay, so there are some medical applications associated with this particular toxin. Okay, what's this? Notice the arms pulled up like this. It's a little bit hard to see, but the back is usually arched. This is because the muscles in this particular disease are basically in a state of permanent contraction, a state of tetani. Okay? Didn't you do something in physiology with that, with the frog leg muscles, and you stimulated them and didn't let them relax, and you got that state of tetani? Okay. Well, this is a disease tetanus. And it's due to a toxin that behaves very, very similarly to the botulism toxin. The toxin is called tetanospasmin, and it causes these spastic contractions of the muscles. They can't relax. The toxins produced by a species of Clostridium tetani, And if the spores are introduced into the body and can germinate, then the tetanus toxin actually migrates, retrograde, backwards basically, up neurons until it gets to the spinal cord. So the toxin actually transfers through the nervous system up to the spinal cord. What's historically the most common ways, or at least is said to be the most common way, to get tetanus? Step on a rusty nail, okay? But really, any type of puncture wound, attack, a thorn, anything that might be contaminated with these soil spores could cause tetanus. And if you think about it, it's kind of a nifty system as far as the bacteria is concerned, because when you get a puncture wound, they're injected into the tissue. When you pull out whatever it is you got punctured by, that kind of seals up the skin, okay? And then as cells start to die because of the trauma, we create that little mini anaerobic environment. The cells start proliferating and producing their toxin, and then that travels up the spinal column. The major difference between the tetanus toxin and the botulism toxin is the botulism toxin affects cells that are at the myoneural junction, the nerve-muscle junction, that cause contraction. They're called excitatory neurons. Okay? There are other types of neurons that also innervate muscles, and those are inhibitory neurons. Excitatory neurons cause contraction, inhibitory neurons cause relaxation. Well, when you flex your arm, for example, don't muscles have to contract and then relax? Contract and then relax? Because they're getting the appropriate signals from the appropriate neuron at the appropriate time. Excitatory, contract, inhibitory, relax. Okay? Well, the targets for these tetanus toxin molecules, the tetanospasmin, is that it migrates up to these inhibitory neurons and prevents those inhibitory neurons from sending the proper shut-off signal, relax, to the muscles. So the muscles are in a permanent state of contraction. So we get that pulling up of the arms. We get the arched back as your back muscles are constantly contracted. What's another set of muscles that characteristically get paralyzed by continually contracting in this particular disease? Jaw, facial muscles. Common term for this particular disease is lockjaw. Okay? So basically the same type of toxin with the same mode of action. This particular toxin is also a peptidase, but it interferes with vesicles being released that contain the inhibitory neurotransmitter, not acetylcholine, which is the excitatory one, but the inhibitory one, which is something like GABA, GABA-aminopentanedic acid, or butyric acid, GABA-aminobutyric acid. So they're neurotransmitters that are both excitatory and inhibitory. And the tetanus toxin interferes with the release of the inhibitory neurotransmitter. So these are a few of the toxins that pathogens can produce. These are some of the virulence factors that cause the characteristic symptoms associated with the disease. Now, sometimes when bacteria produce toxins, they just secrete it out into the environment. And basically, it's kind of a random chance whether the toxin will bind to its particular target cell. Some bacteria have developed toxin secretion systems which basically inject the toxin into the target cell. As you might appreciate, this would be more efficient, and there would also be less risk of diluting the toxin by just sending it out into the body fluids. If we had all kinds of time and had the inclination, we could spend a fair amount of time talking about secretion systems. This particular slide just gives you some examples of different types of secretion systems that have been identified for various bacterial toxins. And we're not going to go through those, but I just wanted you to be aware of the fact that this is a strategy that can be used to try to be more smart about delivering the toxin into a particular type of cell. So those are all exotoxins. We also have endotoxins. Endotoxins are not protein molecules. Most of the exotoxins are proteins. Instead, these are lipid A molecules that are part of the LPS layer of gram-negative bacteria. These are liberated when the cells lyse. Lipid A is a type of molecule called a PAMP. And I'm going to introduce that term now, and then we'll come back and talk about it later on when we talk about immunology. PAMP stands for pathogen-associated molecular patterns. A PAMP is kind of a molecule that says this cell really doesn't belong. It's something that normally is found only on pathogens. We normally shouldn't have those as part of our cells, and therefore this is something that we would mount an immune response against. And there are certain types of white blood cells, for example, that recognize these PAMPs. And when they see them, they latch onto those cells and then carry out a set of immune responses that results in elimination of the cell. Activation of immune cells by PAMPs can cause fever, inflammation, change in vascular permeability, so that can lead to fluid leakage. Fever is typically one of the hallmarks of endotoxin poisoning. Exotoxin poisoning typically isn't associated with fever. So if you've ever been unfortunate enough to have salmonella food poisoning, salmonella food poisoning is the result of endotoxin poisoning. Salmonella die when they're in the food, release their LPS layer, and then if you ingest that lipid A, that can ultimately cause an immune response which gives you the symptoms of food poisoning. Now, not all food poisoning is the result of lipid A. Okay? Staphylococcus aureus can cause food poisoning as well. Does it have lipid A? No, if it's a gram-positive bacteria. Okay? So it produces some toxins instead that have an effect on intestinal cells. Endotoxin poisoning usually isn't lethal. Although if you get a bad case of food poisoning, sometimes you wish you were dead. Okay, it's pretty nasty. A lot of diarrhea, vomiting, okay, a lot of intestinal effects from this particular response. In infants, it can be, so you have to be careful. When we vaccinate against diseases like diphtheria and tetanus and pertussis. What's pertussis? Whooping cough. Okay, they're all caused by exotoxins. What we can do is we can administer an inactivated form of that particular toxin and then immunize with that to mount an immune response, basically to educate your immune system. This is what those toxins look like if you ever should be exposed to them. These particular toxins are proteins, and proteins are very strong antigens. What's an antigen? Pardon me? No. It's something that an antibody recognizes. It's something that stimulates the production of an antibody. Antigen stands for antibody generating molecule, or antibody producing molecule, if you like. Okay? Typically, we think of antigens as being foreign substances. Okay? And if they're introduced into your body, you will mount an immune response against those antigens by forming antibodies. Okay? So that's why we can vaccinate. We can develop inactivated toxins by either heating them. There are various chemical treatments, including formalin, that you can use to kind of denature the toxin, but it still maintains its antigenicity. So it doesn't harm, but it does elicit an immune response. Okay? Lipid A and lipids in general are very poor antigens. And that's why there really probably will never be an effective vaccine against endotoxin poisoning caused by gram-negative bacteria. They're just not good antigens. They're not going to stimulate an immune response. So here are some comparisons with endotoxins and exotoxins. This will be a nice summary for you when you're comparing those two particular diseases. Note down here specifically from a diagnostic point of view, fever stimulation is typically associated with endotoxin, but not with exotoxin. One of the things you've really got to be careful about if you're a physician is if you admit a patient into the hospital with a septic infection. What is septic infection? It's in the blood, okay? And that's serious. And it's caused by a gram-negative bacterium. What's going to potentially happen if you give them big doses of IV antibiotics to try to treat the infection?
They all die. They all die at once.
They're all going to die at once. When they lyse, they'll release their lipid A, and so you can cause a really massive release of endotoxin. So, very difficult situation when you have septicemia. Sometimes your treatment causes additional problems. Third set of virulence factors are the antiphagocytic factors. And the antiphagocytic factor that we've talked about most are the capsules. Capsules make it very difficult for white blood cells to phagocytize the pathogen. Sometimes it's been analogy's been made is having a capsule is almost like that slippery bar of soap you really can't handle and you really can't grab onto. But there are some other strategies that allow pathogens to actually survive within the phagocyte. During the process of phagocytosis, the white blood cell ingests the pathogen. Okay? It surrounds and engulfs the pathogen. And it traps the pathogen in a vacuum. That vacuole is called a phagosome. And then the phagosome can fuse with a lysosome and form a phagolysosome. Now, typically what you expect will happen in the phagolysosome is some killing mechanisms will be activated. And so the digestive enzymes from the lysosome will start chewing up the bacteria. Remember within the phagolysosome that there is an increased oxygen uptake and there is an increased production of some of those reactive oxygen species, and that contributes to the killing of the pathogen as well. But some microorganisms have developed the ability to survive within those phagolysosomes. Well, that's a pretty good deal for them. Because now, when you're in the phagolysosome inside the white blood cell, what are you protected from? Other white blood cells, antibodies that might have been secreted, okay? And white blood cells migrate. And one of the places that they migrate to routinely are lymph nodes. And so these particular bacteria can accumulate in the lymph nodes by these white blood cells that carry those bacteria migrating into those locations. Eventually, the white blood cell will die and the bacteria can be released. And some of them even have mechanisms that kill the white blood cell so they can then be released into the lymph nodes. Tuberculosis has a tendency to do this, to be able to survive within phagolysosomes and then be spread to different locations within the body.
So my doctor always checked to see what's wrong.
Well, yeah, swollen lymph nodes are in general an indicator of infection. Because when we talk about the immune response, we'll see that white blood cells can bring pathogens or pieces of pathogens to other white blood cells that reside in the lymph nodes and activate them to start an immune response. And part of the activation process is those white blood cells in your lymph nodes start to multiply. And they increase in numbers, and that's why your nodes swell. So palpating your lymph nodes is oftentimes an early check to see if they're swollen. That would indicate infection. Okay. Best way, though, to indicate infection is get a blood draw and actually see if certain populations of white blood cells are increasing over what they should be normally. So that might be a follow-up test the physician would do, is to get a blood draw. Staphylococcus aureus is a pretty nasty pathogen. One of the reasons that it's a nasty pathogen is it can have multiple virulence factors. And they can fall into all three of the categories that we've talked about. We're only showing two of the categories here, either exoenzymes or toxins, exotoxins. But these can also produce anti-phagocytic factors as well. Look at the long list of effects that can be the result of the liberation of these particular toxins. Okay, we talked about coagulates and what that does. We talked about hyaluronidase. We talked about staphylokinates. They can produce some lipases. Many of those species of staph are resistant to penicillin, so they produce penicillinates. Hemolysins, enterotoxins, exfoliative toxins cause desquamation of the skin. The skin just starts to kind of peel off in layers. We mentioned the toxic shock syndrome toxin. Staphylococcus aureus can cause three major categories of diseases: non-invasive, cutaneous, and systemic diseases. The major non-invasive disease that they're responsible for is causing certain types of food poisoning. That's really one of the more common forms of food poisoning. Fortunately, food poisoning is fairly self-limiting. Symptomology usually only lasts about 24 hours. It's got a pretty rapid onset, usually about 4 hours after ingestion of the toxin that they produce. It's enough to start producing symptoms. As opposed to the endotoxin type of food poisoning, and this is actually called technically food infection, not food poisoning. You don't get the fever associated with this. So that's one of the ways to differentiate between the cause of your food poisoning. There are a number of cutaneous infections associated with Staphylococcus aureus. Here's an unpleasant looking one called scalded skin syndrome. And this is the result of the production of that exfoliative toxin. And you can see that the skin is starting to kind of peel off in sheets right here. You may know what this one is? This is another staph infection. You get it? What it's called? Impetigo. Yep. Kids are kind of notorious for getting impetigo. You can also get basically impetigo, same kind of thing, associated with your eyes. You know what that's called? You may ever had a sty. Okay. That's basically impetigo of the eye as opposed to the skin. There are other diseases as well of the skin. You can get boils. These are boils. That's a staph infection. There are also things that are related to them called carnuncles and furuncles. But they're all basically inflammatory diseases of the skin caused by staph. Most acnes are bacterial in nature. There are several different types of bacteria. In this particular example, it is not staph that they're showing, but another type of bacteria called Propionibacterium. But basically what they do is they infect the hair follicles or pores in the skin. And that dead accumulation of cells that results from the toxins that these pathogens produce can cause whiteheads. Later, as tissue starts to die and necrose, blackheads. And then you can get a pustule that forms. Okay? We used to think that acne was primarily caused by diet. And when I was growing up, the mantra was, don't eat greasy foods, don't eat chocolate, because they cause acne. It's a bacterial disease. Okay? It's an infection of particularly hair follicles and pores in the skin, and it's an inflammatory response. So this is just a summary slide of some of the types of diseases that are caused by acne. Some of these systemic diseases include the toxic shock syndrome. I already mentioned that. Toxic shock is a superantigen. And it stimulates immune cells to start producing a lot of those cytokines, the cytokine storm, which results in massive levels of inflammatory response and can be lethal. In the early 1980s, there was an outbreak of toxic shock syndrome. And a lot of women seemed all of a sudden were being brought to the hospital with severe cases of toxic shock. This is where epidemiologists really were important. Epidemiology, we're going to talk about that in a little while, is basically the study of the causes and distribution of diseases. They're kind of medical detectives. And what they were able to ultimately figure out is that there were certain brands of tampons that were very super absorbent, and they created ideal environments for those Staphylococcus aureus bacteria to grow, and then they were pumping out massive amounts of this toxic shock toxin. So super absorbent tampons were then withdrawn from the market. Then Food and Drug Administration required tampon labeling, and as those came off the market and stopped being used, you can see that the incidence of toxic shock syndrome plummeted dramatically. Toxins can become systemic and affect the bone, and you can develop a debilitating condition called osteomyelitis. Toxins can affect the endocardium surrounding the heart and cause endocarditis and inflammation of that. Anytime that there are these systemic diseases, it's a problem. Endocarditis, for example, is a 50% mortality rate. So that's pretty variant. You've all heard about MRSA. MRSA actually kills more people in the United States than HIV does. Pretty serious disease. They're working right now on trying to develop a vaccine against Staph aureus. Okay? It's a serious enough pathogen that public health officials believe that would warrant vaccination if we develop this vaccine for it. Okay, well, Thursday we'll talk a little about some of these strep pathogens and some of the diseases that they cause as well. So again, make sure that you have a good amount of growth on your unknowing. We're going to work in groups of 3 again in terms of inoculating knowns and unknowns. And this is the last big batch of inoculation of your unknowns. Okay? Most of the follow-up testing of this will be done on Friday. So some of these tests will require adding certain reagents to actually be able to interpret the results.
Not this Friday, but next Friday, on the 20th.
Okay?
I have a family wedding to go to in
Okay.
Minneapolis, so it's going to be Friday morning.
Okay.
I already looked forward in the lab, and we're just basically taking over what we did Tuesday.
Okay.
So would I be able to come in Thursday?
You should be able to,
look
yeah.
over it?
Yeah,
and
you
then
should be able to, yeah.
Will I miss any participation points or
No.
or not? Okay, thank you.
Oh my god.
You're like me, huh?
My back kills.
So where's it located? In the middle, side?
L5 and L4, bro.
Great.
Not fun at all.
Good back.
I was just complaining.
Neither one of us have good backs.